Welcome to Soft Genetics webinar, Fragment Analysis in 2025, Applications, Challenges, and Smarter Workflows. I'm very excited to share information on the wide range of applications, challenges, and efficient workflows with you today. And I'm happy that we have attendees from a wide range of molecular biology fields. Before we start, I'd like to introduce myself to the group. I've been with Soft Genetics for about 17 years in the DNA fragment, forensic development, and support team. Prior to joining Soft Genetics, my work experience included hospital and research labs ranging from molecular disease resistance research in agriculture to primer design and data analysis in forensic and clinical applications. One of the most enjoyable parts of my work at Soft Genetics is interacting with lab scientists to learn what tools are needed to improve their data analysis workflow. Thus, the title of today's webinar, Applications, Challenges, and Smarter Workflows. Fragment analysis has been a core part of genetics for decades. Today, we're going to look at why it's still going strong, what challenges labs face, and how modern tools like GeneMarker and ChimeRMarker make things faster, easier, and more reproducible. I plan on covering a series of topics, why fragment analysis still matters, some key applications and trends, common workflow challenges, and then smarter tools in GeneMarker and ChimeRMarker software to help with those workflow challenges. I'll present a live walkthrough, one in GeneMarker and one in ChimeRMarker, and I'd encourage you to please put any questions you have in the chat box. I'll respond to any questions that we don't get to during the presentation via email later. So let's take a few minutes to talk about the importance of fragment analysis in many different fields. These two timelines track the evolution of sequencing in the upper figure and fragment analysis in the lower figure. Both sequencing and fragment analysis relied on slab gels and radioisotope labels in the early years. In the 1990s, capillary electrophoresis and fluorescent labels improved workflow for Sanger sequence analysis and fragment analysis. Fragment analysis also leapt forward with AFLP that utilized fragment restriction in combination with PCR to produce the unique fragments that would be separated by slab or CE. Sequencing via NGS, next-gen sequencing, and long-read chemistries provides detailed data that was unheard of 25 years ago and is the cutting edge of many research and medical testing areas. In the mid-1990s, SSR, STR, and microsatellites became workhorses of analysis for many fields, including genetic diversity, forensics, paternity testing, population genetics, and medical clinical research and screening applications. Fragment analysis is starting to be accomplished via NGS for fields such as forensics, chimerism, bone marrow transplant, and detection of copy number variants in cancer and aneuploidy cases. I do want to pause to mention that even though NGS for these applications provides additional information, such as STR isoalleles, the positives obtained from copy number variation screening in NGS are confirmed via fragment tests, such as MLPA with CE separation of fragments. Each technology is built on the strengths of its predecessors, expanding capabilities in fields as diverse as population genetic diversity, forensics, and medical and clinical research, and aiding in diagnostics. The take home from this timeline discussion is that while sequencing gets a lot of attention, fragment analysis remains the gold standard for a surprising number of applications, from STR profiling to MLPA and chimerism and other clinical tests, and to plant and animal genotyping, diversity research. Why? It's fast, it's cost effective, and importantly, validated in regulated settings. This is a summary, although it's not inclusive, of some of the core applications: STR and human ID, microsatellite genotyping, microsatellite instability and loss of heterozygosity, plant and animal breeding, MLPA, MSMLPA, copy number variant screening, and methylation detection and imprinting, and AFLP and SSR marker analysis. These aren't fringe applications. They're, they're These aren't fringe applications. They're foundational to human identification, biodiversity tracking, and copy number quantification. Fragment analysis is often the right tool when sequencing would be overkill or too expensive for routine use. STRs are the basis of combined DNA index system, or CODIS, for forensic database searches for crime scene data to samples such as missing persons and convicted offenders. While MPS sequencing is starting to be used in the forensic applications, the majority of samples processed are as STRs separated by capillary electrophoresis. Even the MPS sequences of STRs are back compatible to be consistent with length-based alleles from decades of CE and STR research. Please see our forensic videos for more details on data analysis tools for forensic applications. Chimerism testing post-bone marrow transplant takes advantage of human STRs and is the most commonly used screening and monitoring process to track chimerism. Human identity STRs provide a cost-effective way to monitor stable chimerisms and for early detection of GVHD, or graft versus host disease. Microsatellite genotyping, AFLPs, and SSRs are powerful tools for biodiversity and ecology research. Microsatellites, or STRs, are essential in plant and animal breeding. Additionally, microsatellites and paternity testing are not limited to human parentage testing. Breeding of livestock and pedigreed dogs and cats previously relied on written records of parentage. DNA paternity and familial testing provides absolute confirmation of parentage, as written records can be manipulated, but the STR pattern of an individual cannot. DNA analysis for microsatellite instability, loss of heterozygosity, copy number variants involved in cancer development are important tools in hospital laboratories, with MLPA and MSMLPA used either initially or as confirmation of results from MPS or next-gen sequencing screening. This discussion is just a tip of the iceberg regarding capillary electrophoresis of DNA fragment applications. Please check out our webinar pages for detailed videos on these topics as well as other applications. And now that we've reviewed the importance of DNA fragment analysis in today's laboratories and some of the key applications, let's talk about the elephant in the room, the pain points where labs struggle to obtain the most accurate and consistent data analysis from each sample. And so we have the problems listed on the left and their impact on the right. Things such as manual binning, which are time-consuming and prone to error, stutter and pull-up artifacts that result in missed or miscalled alleles, batch normalization, drift causes inconsistencies, and tool complexity. There's often a reliance on in-house scripts or outdated systems for post-genotyping analysis. Sound familiar? These are real issues in many labs. Even experienced users spend hours correcting allele bins or rerunning plates due to migration drift. We'll take a look at examples of each of these pain points. Manual binning is essential for AFLP and early microsatellite studies, ranging from microbe diversity and epidemiology to etiology. While some STRs have simple sequence repeats and consistent stutter artifacts, other STRs have complex sequence repeats that result in LUS, or longest uninterrupted sequence, that requires more specific stutter filtering than traditional analysis methods. Despite using a size standard in each specimen, CE drift from different experimental conditions or different CE instruments requires panel calibration that is time-consuming if performed manually. Lastly, for the purposes of this discussion, reliance on in-house scripts or outdated systems is not only time-consuming, but also prone to human errors when results from the fragment analysis software are transferred to macros for post-genotyping analysis. GeneMarker and ChimeRMarker were built specifically to handle these problems. They are used globally in clinical, wildlife, agriculture, and forensics labs exactly for this reason. To further explore this, I'd like to take a look at examples of some of the issues and the solutions provided by GeneMarker and ChimeRMarker software. Looking first at auto-binning, we have a screenshot of an AFLP panel. On the lower right trace overlay, you can see that there are many peaks that are exactly the same size, but there is a sample where the allele is shifted to the right. Auto-binning in the panel editor detects peaks with slight shifts and extends the bin to the appropriate width, providing consistent allele calls from one analyst to the other. Built-in mixture deconvolution in ChimeRMarker software greatly increases analysis efficiencies, up to 70% in one publication, and it increases laboratory throughput per an associated molecular pathology presentation from the Children's Mercy Hospital. This is accomplished by providing the software with donor and recipient genotypes. The software automatically assigns donor, recipient, or shared origin to each allele in the mixture, instantly evaluating which are informative markers, as is seen in the upper figure. The chimerism application applies best practice guideline calculations to the deconvoluted mixture. This streamlines the analysis pipeline from genotyping to chimerism, and quality metric calculations in the lower figure is an example of a single time point. The application also has long-term monitoring reports. Automated panel calibration and selection of the best allelic ladder for each sample is one of the best solutions for migration shift that causes issues with batch processing of data analyzed with human identity chemistries in applications such as chimerism, maternal cell contamination, and forensics. In this image, we see 3 samples and 2 high-quality allelic ladders at the bottom. The migration shift is quickly seen in the MLPA with the first allelic ladder shifted to the right. After calibrating the panel to the allelic ladder FSA files, the software compares each high-quality allelic ladder to each sample and selects the one that most closely matches the migration pattern of the sample. In this example, each sample electrophoregram lists first the file name of the sample, followed by the reference ladder used in parentheses. The auto-calibration and selection of the best ladder reduces analyst intervention to apply the appropriate ladder. It's more accurate than panel averaging, and it allows data from different dates and or different CE instruments to be analyzed in the same batch. One of the requirements for ISO and CLIA compliance is the audit trail. User management with password-protected analysis rights provides. One of the requirements for ISO and CLIA compliance is an audit trail. User management with password-protected analysis rights provides a full audit trail of any flags and interventions by the analyst. The last item for this section of the presentation is the embedded applications. The software is applicable for STRs, SNPs, MLPA, and CNV, whether in environmental data analysis for things such as AFLP and quantitative analysis, clustering for dendrograms, or relationship testing and database searching in wildlife forensics and purebred livestock confirmation, or clinical applications such as MLPA, MSMLPA, snapshot, MSI, trisomy, LOH, or chemistries for specific diseases, Fragile X, cystic fibrosis, or repeat expansion diseases such as Huntington's disease. These embedded applications for post-genotyping applications simplify the workflow, increasing productivity and decreasing potential for data transfer errors. Advanced labs can use custom panels, repeat-aware quality control filters, and even automate the detection, filtering, and rare allele flagging. If you're currently exporting Excels or running macros for post-genotyping applications, you don't have to anymore. At this point, I'd like to take a few minutes to look at some of the solutions we've discussed and the streamlined workflow of genotyping in two different post-genotyping applications, one in GeneMarker and one in ChimeRMarker software. To start, I'd like to do a quick open data, and we'll look at some plant microsatellite data. Okay, these are SG1 files. More commonly, these would be FSA files. Okay, they are from Eucalyptus STR multiplex. Once the data are imported, you can see the raw samples here, evaluating those if you wish, okay, or go right to the analysis. The lab can make different templates. These are microsatellite dinucleotide repeats. Okay. So we'd select the microsatellite dinucleotide repeat application or template, which has the panel, the size standard, the fragment type. In the second screen, there are a variety of data processing options that are included in the template. And then finally, we have just the peak score evaluation, and we say OK. Okay. After analysis, the analyst has immediate indication of accurate size calling, as can be seen by the green sheets here on the left. This indicates that the size calling is linear and high quality. There is a size calibration option here. For additional details, the size calibration provides information about sizing accuracy, comparing the migration pattern of each sample to the ideal migration pattern, linearity, and any migration variance, as is typical for this 250 base pair fragment in GeneScan 500. If there are issues with the internal lane standard, it is possible to manually intervene and rescue samples. Please refer to our how-to guides from the resource dropdown on the Soft Genetics website for a detailed video on how to accomplish this manual calibration. Okay. Back in the main screen, the analyst would evaluate any of the calls that fired analysis rules with yellow or red flagging, as the green flags indicate that the peak passed all of the quality filters. The first yellow one that I come to is down here at up to 329 base pair fragment. Double click on it, and it goes right to that section of the electrophoregram. We see the peak has a red underscore, and the table underneath indicates that this was saturated and repaired. Okay, one of the views that will help in evaluation of this is the all-color view, which has crosshairs when we can put it directly over top of the saturated peak and see that although there is some spectral issue, it's not causing any artifact peak calls. And so the analyst would come in and confirm this. They would delete the artifact peak there and confirm this saturated peak. This is all then part of the audit trail, as we see that it is recorded that it was a quality reason of saturated repaired, and that this was confirmed then by the analyst. So at this point, many labs would be exporting the genotypes for further analysis in relationship testing or cladogram software to produce dendrograms. GeneMarker has an extensive selection of post-genotyping applications. These applications were developed at the request and with the feedback of end users in clinical and agriculture, wildlife, labs. The embedded applications provide more streamlined workflow and remove the need to export from genotyping to secondary analysis software. GeneMarker is also contained in the lab's computers, reassuring for data security that no internet connection is needed. For the purpose of this demonstration, let's look at cluster analysis. When we select the cluster analysis, it opens the cluster analysis settings, and you can choose from the various settings. I have it here set at Euclidean distance, but let's say we wanted to look at similarity and percent same genotype, we'd make that change in the setting and the cladogram will update. Replacing the file name with a more descriptive name is available from an editing tool in the application, and the image can be exported as a PDF for presentations or publications. Clustering analysis is just one of the many post-genotyping applications. Please refer to videos of other applications on our main webpage. Next, we'll take an overview of ChimeRMarker. So next, let's take an overview of chimerism evaluation and monitoring post-genotyping that is routinely done using human identity PCR multiplexes to monitor chimerism status. ChimeRMarker software has the same user-friendly interface and rigorous pattern match size calibrations that we just reviewed in GeneMarker. In addition, ChimeRMarker uses the genotypes of donor and recipient to automatically label the origin of each allele in the post samples. Each peak has a designation for origin, D for donor, R for recipient, and D/R for peaks that are shared by both the donor and the recipient. These labels are consistently identified in all samples, removing the time-consuming and potentially error-prone step of identifying informative loci manually. Looking at the project from left to right, the file name tree at the left has the raw data folder and the allele call data folder. The allelic ladders, negative and positive controls, were evaluated during genotyping. Origin designations are seen in the electropherograms. As we see here we have an electropherogram of a recipient, a mixture that's at about a 9:1 ratio, and a donor, and these are all in the FAM channel shown here. You can see the flags. Just kind of zoom in a little bit. And the origin is also reported in the allele table at the right. As with GeneMarker, the analyst would review the allele calls prior to accessing the post-genotyping applications. The chimer panel is specific to the two individuals in the case. In this brief section, we can see two partially informative loci, amelogenin and D3S1358. These have a shared peak and a unique peak for one of the single-source samples. Two other loci in this screen, D1S1656 and D2S441, are examples of fully informative markers with unique alleles for both the recipient and the donor. This time-saving step is the main feature that results in laboratories significantly increasing productivity and efficiency. From the post-genotyping application, you see that only the single donor chimerism analysis is available. And that's because the panel is one that can detect only one donor, and the maternal cell contamination is also grayed out. Okay, so we select the single donor chimerism. Okay. These settings can be locked in with user management to ensure the lab SOP is followed. I've selected a more stringent setting without deconvolution here to mirror the best practice guidelines to use fully informative loci whenever possible. The chimerism analysis screen lists each sample. Let me get to that screen. Okay, so the chimerism analysis screen lists each sample on the left, the result table in the center with the results for percent chimerism, locus error, measurement error, limited detection, and whether or not the locus was usable in the table, and then a summary at the bottom. So here we see average chimerism 9% plus or minus 4 standard deviation. And this particular sample is a 1 to 9. So it's using here peak height ratios. Depending on the requirements of your limb system, you would either export the table here by using the save icon, or you could go to the printer preview to select a PDF, an image report. For each sample, there would be a header with specific information regarding the analysis and the analyst. The electropherograms, here we see the first 3 dye channels. Any of the markers that were not used per the settings have a red highlight. The second page for the sample is just the red trace that didn't fit with the other ones. And then finally, the report table per marker and the summary. These can be saved then and as either PNG images, JPEGs, or PDF images. There's also an option for graphing of long-term monitoring, and also per long-term monitoring, if we return to the main screen, if a project is reopened, there is an option to add additional samples so that once a project is made for a case, any new samples can just be added to that project and analyzed. Okay, so this is in summary, this is an application that greatly enhances the workflow and the accuracy for chimerism monitoring. So in summary, GeneMarker and ChimeRMarker have many advantages. Let's break it down into a few groups. Time studies indicate up to 85% faster analysis in some cases, and increased analysis capacity of 20 to 30% annually. This is from a 5-year study using ChimeRMarker. Please go to our website or contact info@softgenetics for these references. The software is intuitive. As we saw from the walkthrough, there are drop-down menus for analysis template selection, linked post-genotyping applications, report settings, and various templates. GeneMarker and ChimeRMarker are easy to use with embedded modules, access to online help documentation, and biologists support scientists. The software has reproducible, lab-validated results in diverse areas ranging from wildlife forensics, genetic diversity, and agriculture to clinical research applications and reporting. So what's next? We're actively building for the future, and the feedback we get from webinars like this directly shapes our roadmap. Areas that we're looking at include integration, how to better integrate with limb systems, PDFs and text reports. Labs, this would benefit labs by increasing their interface capabilities. Automation, we're looking at auto-run interface for batch processing and audit trail export without technician intervention. We're also exploring the potential for cloud-ready version and IVD versions. Very importantly, we value your lab's suggestions. Please enter suggestions into the chat or send them via softgenetics.com/contact us. Are one of these 3 areas of more interest to you than others? The IVD versions, automation, or LIMS integration? Or are there areas of interest to you that were not included here? We look forward to hearing from you. One of the most efficient ways to evaluate the software and determine if it will improve your data analysis workflow is to try it out. SoftGenetics has a link on the main page to download free 35-day software trials. Thank you very much for your interest and attendance to this presentation. Please contact us with any specific questions or visit our website for more information.